Hello everyone, welcome to my channel. You might have seen me doing so many tutorials on structural bioinformatics, drug designing, Genomics, quantum physics, siesta, etc. Some of my subscribers told me that why not do some tutorials on R programming and Python programming as well for bioinformatics analysis. So I thought of start by making some tutorials on R programming, especially R programming for bioinformatics. So today we are diving into one exciting and incredibly useful topic in the world of data science and bioinformatics, that is R programming. So basically what we are going to do is we are going to use different packages from Bioconductor, and we are going to install many bioconductor packages which are useful for bioinformatics analysis, such as RNA sequencing, single cell RNA sequencing, even proteomics analysis, mass spectrometry analysis, etc. But one thing that I would like to say, recently I have watched so many YouTube videos where they tell you about how to set up our environment and how to use the packages in a specific environment so that you can transfer to another computer, or maybe later on if you want to try those packages will be stored in your R environment and you can use it anywhere in any operating system. But there are some limitations in doing that that might cause some inconsistencies in using those packages and utilizing those packages for bioinformatics analysis. So I thought of doing tutorials on one of the best packages that R has developed recently, that is called Quarto package, and this is one of the best for documentation, reproducible research, whether you are a seasoned data scientist or student or someone interested in reproducible research and technical publishing. So you will find this Quarto tool very extremely helpful. So Quarto is an amazing tool that integrates seamlessly in R, allowing you to create high quality reports, presentations, and even interactive websites as well. It's a game changer. You don't have to set up our environment, or you don't have to go for downloading the specific packages from Bioconductor and use it when you shift to another operating system, or maybe later on if you want to utilize those packages. That might not be optimal if you use you can use our environment, but sometimes it might cause some problems. So the R community has developed this one of the best packages that I've ever seen, that is called Quarto package, where it can create your R environment automatically. You don't have to do anything, and it can render the documents very nicely, very neat and clean, so that your colleagues, your students, or your professor can read the documentation and start analyzing all the the data, the bioinformatics data. So by the end of this video, you will have a solid understanding how to set up a Quarto project from scratch, how to write, render documents, and some best practices to make your projects stand out. I'll walk you through every step of the process, ensuring that you can follow along regardless of your experience level. So if you are a beginner, if you are an expert, you can follow my tutorial. That will be extremely helpful for you guys. So for the later development or the advancements, I would recommend everyone to have some basic statistics knowledge, because we are going to dive into many different packages in R, and we'll be covering mostly all the aspects of DE-Sec2, stars, single cell sequencing packages. Nextflow will be covering variant calling, and even mass spec, proteomics, maxquant, lemma, DEP package, all those things. So we'll be mostly using bioconductor packages and different R environments as well. So we'll be using Quarto for our documentation, and this will be incredibly helpful for you guys to understand the concepts of R. And yeah, so if you like my videos, if you have not subscribed to my videos, please like, share, comment, and subscribe to my channel so that I can make this kind of tutorials for you, and I hope you like it. Thank you. So let's begin. So the first thing that you want to do is you have to visit this particular website where you can download the R and the RStudio. If you click on this link, it will be redirected to this particular link where you can download R for your specific operating system, for Linux, for Mac OS, and for Windows. I am using Mac OS, so I'll be downloading this one. So I have already installed R for my Mac. Okay. So after installing R, then you go for the install RStudio option for your operating system. I have already downloaded the RStudio for Mac OS, and this will install the latest version of RStudio, which contains different kinds of tools and latest packages. Okay, so once you install this, so we'll be covering the Quarto documentation. This is the website. I'll put the link in the description. So this is the Quarto documentation. This is one of the best technical and reproducible documentation for scientific purposes. In our case, it's for bioinformatics analysis. This not only works for R, but also for Python, Julia, and other different softwares. It has also a VS Code extension as well. And if you want to get started with the Quarto project, you can click this guide option and using R, and you can see different types of examples, how you can set it up, and what all things that you require for this to have a nice presentation or documentation for your project. Okay, so let's dive into RStudio. So this is the graphical user interface of RStudio. Many YouTube tutorials I have seen, and they start with creating a file called RScript. Okay, so whenever you start a project, they tend to create RScript and they write the code over there, and they execute those commands in the console. But we are not going to do that. We are going to set up a project directory first, and then we run all the analysis. So before delving into setting up a new project directory, I would like to tell you about the RStudio background information. So this is the graphical user interface where it has a console, it has environment, and it has another file directory. So all the plots that we'll be generating will be displayed over here. These are the packages that have been already installed or have been loaded into your R environment. Those packages have been listed over here. The help option is best. We can get into this when we try to run a calculation using RStudio packages. I can tell you how you can look into the help documentation over here. And then the viewer option is for the documentation. If you have a PDF or a Word document, and this is the viewer option where you can render all your codes, etc. And this is for the presentation purposes. Okay, so this is the environment, and the commands that you are going to execute over here will be displayed over here. I'm going to clear this out. Okay, all the history of all the commands that you have executed. This is the connection tab. If you have a server connecting to RStudio server, then you can utilize this connections option. And these are the basic tutorial option you can learn from by clicking here. Okay, so the basic thing that we focus is console, the environment, and the files and packages and the plots. And in here you have the console where your R code will run, and you have the option of terminal as well. Like you open CMD terminal in Windows and Mac OS terminal. So basically you can execute the commands over here as well. So this will act as your terminal inside RStudio. Okay. And if you are running some analysis that requires some background jobs, background running jobs, so it will be displayed over here. Okay. So now I'm going to tell you some of the basic setup of RStudio. So first thing first, what you want to do when you install RStudio and open this particular environment is go to the tools option and then go to global options first. Okay. If you want a dark background like the theme that I have installed, or if you like a light background, if first you go to the appearance option, and here the option editor theme, you can select any type of themes that you need. So these are the themes I have installed externally, but you can choose whatever the themes you like. You can set the font size. I have set the font size to 12. The default font is Monaco, which is the default one for RStudio. You can choose whatever you like. There are many options, Arial, Roboto, or whatever the fonts that have been installed on your PC will be displayed over here. You can zoom it and you can select the theme as well. Okay. The next thing that you want to go is the general option where you want to work. So generally what I do is I basically set up the home directory because it's the best option to run any kind of this type of programs from the home directory itself. So for the Mac OS, it's the best option to choose the home directory, and you run different types of packages from there, or your bioinformatics analysis from there. Okay. Then you go to the code option, and here there's a option called display where you can highlight different types of options like selected word, selected line, line numbers, etc. And if you want to make it fancy for your RStudio, so this is the option where you can modify based on your choice. Rest of the things we don't want to touch right now. Oh yeah, there's also another option called pane layout. This is the default one. They have 4 columns. One is source, console, environment, and these files, plots, and packages. But you can set up as per your wish. Okay. Once you select different options, then click on apply, and then okay. The next thing that you want to do is set up a new project. Okay. So for that, what you want to do is go to the file option, go to the new project, and create a new directory, or do it from your existing directory. So I'm going to create a new directory, and then in here you don't want to select any other options like our package, Shiny application, Quarto. You don't want to select anything, just new project. Okay. And give a directory name, something like bioinformatics analysis. So you can see that I have given this underscore option. So all the bioinformaticians out there, all the data science specialists, those who are working on file namings, they always, for the multiple words, they always put underscore in between, so that if you want to access it from the terminal, then it will be easy for you to access this. Although you can also don't want to set up capital letters, you can just set up small letters for your analysis purposes. So this will be the directory. I'm not choosing this option because we are not delving into the Git repository. If you have a Git repository and if you want to submit your R analysis over to GitHub or something, then you can click this option. For now, I'm not clicking it. We are not going to use that for now, but we are going to use the R environment with this. This option is crucial because whatever the packages that you are going to install in this particular session will be locked in this project. Okay. Now then create project, and you can see that the files, the required packages will be installed here, and you can see over here your folder has been created or your project has been created, such as bioinformatics analysis. Okay. And also you can see that automatically all the files will be created over here. One is R profile, your R project, your R environment where all the packages will be installed, and your renv.log means the version of the packages that you are going to install will be logged in this particular directory or in this particular project. Okay. The next thing that you want to do, since we are going to talk about Quarto project, the next thing that you are going to do is click here this option, plus option, and then go for the Quarto document. You don't want to select any other options. There are many options for R HTML, you know, Python script, R Markdown, R Notebook, but I would recommend all of you to go for Quarto document because it's easy and the visualization is much more impressive, I would say. So go for the Quarto document. You give some title over here, since we are going to do one project here. So let me just name it as project 1, and author is my name. So title is project 1. And you can select HTML option, you can select PDF, and you can select Word as well. But I'm going with the HTML because you can share the link with anyone. PDF, it might or might not work if they have installed some other version, but HTML works for any kind of mobile viewing, any kind of browser, it will work. If you want presentation, then you can go for the presentation like PowerPoint over here. You can go for that. And the basic engine that you are working, the bioinformatics analysis that you are going to do. So the default one is NITR. NITR is for the R, and if you are writing Python course, then it will be best to choose the Jupyter version. So we are going with the NITR, and here it is automatically selected, which is use Visual Markdown Editor. I'm going to tell you what is Visual Markdown Editor. Okay, now you create this. So this particular thing will pop up where it has written that the title, the author, and the format is HTML, the editor is visual. So there are two options. One is source, one is visual. If you click on source, then it will show you all the what I can say is all the codes that you are using. For instance, if you see the difference over here for the visual, you can see that you just have to put the curly braces over here. But if you click on source, you have to put this particular character, and then only it will run. So this is the basic difference between our script and Quarto documentation, because it makes easy to write like you are writing in your Word or Notepad or something like that. So it properly documents everything. So I would recommend everyone to install this, not install this, to utilize the Visual Markdown code, and it will run perfectly. And in here you can see that these options are here. I mean, these are the headers, so you can also put some headers, and it can show all the headers over here. So this is your basic chunk where you write your code, and this is the execute options. So if you click on here, it will be executed. You can see 1 + 1 is 2, so it's perfectly executed. And you can set up echo false where you don't want your output to be displayed over here, but I would not rather say that don't use this echo option. It should be there, otherwise it will be very not difficult, but for the basic purpose of analysis to know what is going on, you can have a clear documentation of that. Okay, so as you can see here, you can execute the code, right? So the next option that you want to look into is this render option. So whatever you write over here, if it is executed successfully, then only the render option will work. otherwise it will run into problem. So basically, before clicking on the render, you have to execute the code perfectly, and then only you can go for rendering options, and then it will be rendered. Okay, you can click this option that if you're writing some code and if you save it, it will be automatically rendered. So that I don't recommend, because every time you save, it will be rendered. It will take time. If you have a big project, big files, so it's very difficult if you render on save. Okay, so if you click on render, okay, here it says what is the file name. So let me project1.qmd. qmd is Quarto markdown files. So any MD files is the markdown files. So this is basically the extension QMD, means Quarto markdown files. Okay. So when you execute that, it will say that some of the packages are not installed. Okay. So these packages you have to install. So what you can do is you copy this install.packages and go to the console. Okay. So the basic command is install.packages, and within the double quotes you write the package name. It will be install. Yes, you click the option yes, so it will be installed. Okay, now you click again this render option, and in the background it will tell you whether it will be successfully executed or not. Now you can see that you have another error, that there is no package called R markdown. So what you can do is you can copy this. Okay, go to the console over here, execute the same command install.packages, but within the double quotes you change the option to R markdown. Yes, and you can see that all the packages that is required to render this Quarto document will be installed. So now you click on render documents. In the background jobs, you can see that it's now executed, and you can see the HTML file has been created and it can show up on your browser. So whatever you do some changes in Quarto documentation, it will be displayed over here. You can see the codes have been here, and this is the output. You can add options to executable code like this, because the previous code that we have shown here, that they don't want to show this, they just only showed you the output. So you can just don't use this and try to render it. Now you can see that your file will be created, and you can see your code as well. So 2 into 2 is 4. Okay, so this is how the Quarto documentation works, or the rendering option works. Now, and this is the option to clear your console. You can clear this. Okay, so now we are going to do one specific bioinformatics project that I mentioned initially, that this is not a tutorial for doing some basic calculations in R, but rather we start with bioinformatics projects. Okay, for that, what we need to do is we need to install some of the packages from Bioconductor. Okay, let me do some modifications over here. So let's give the title FASTQ analysis. Okay, because we are going to do some, we are going to download some FASTQ files, and then we analyze the metrics of this FASTQ file. A simple project, which is the basic one for any next generation sequencing specialist. Okay, so it will be FASTQ analysis. And remember, all these titles and the author should be in the double quotes. The format, the editor, it should not be in the double quotes. Otherwise, if you write anything on double quotes, it will be displayed during the rendering option. Okay, so you can read the documentation, how you can modify, how to make it impressive for your visualization purpose. You can read the documentation, but these are the basic requirements. Okay, so in here what you can do is you can change FASTQ file. Okay, FASTQ file, or FASTQ, or here you can say read the FASTQ file, something like that. So these are the headers. The headers will be displayed like the headers, and below that you have the contents to write. So let me write here. We have downloaded FASTQ file and try to load in R by downloading some packages from Bioconductor. Basically, these are the documents. So you have seen in many editors that if you have to write something, you know, any comment, then you have to put hash. Otherwise, the code will not be executed. It will throw an error. But one of the good thing about this Quarto document is you can write like you are writing in your Word file. So which is pretty cool, right? So this is the advantage of Quarto documentation, not with the R script or not with any kind of editors, but this is the most helpful option. Okay, now we can download some packages and then you know, run some codes or load the FASTQ file and do some metrics or blah blah. Okay, so we will be doing some QC in metrics for FASTQ file. Okay, and this is the chunk. So this chunk you can actually, if you want to install something over here, if you want to write some code because you are downloading some packages from Bioconductor, what you can do is here you go to the insert option, there is an executable cell, and you can click this R option, or you can use option command and then I. That will be for the Mac. I think for Windows it will be Alt, I think Control and I guess. I don't know about Windows, but maybe that will be the option. So you can, but you can go here, insert, and then insert the R executable. You don't want to, you know, put any other code, but only the R executable, right? So now, for the basic analysis of FASTQ file, we are going to download something from website, from any website. So FASTQ file download, you just write in the Google, and here you find some FASTQ files. I'll put the link in the description. So we'll check with this 2.4 MB FASTQ file. We just download this one. I have already downloaded this file, okay, FASTQ file, and I renamed it to sample.fastq. Okay, and remember, once you download this, you have to keep in the directory where you are running. So here you can paste your sample.fastq file, because in this directory you have to put all the raw files that you are going to analyze, and then you can run your analysis. Okay, good. The next thing that you want to do, so you have downloaded the file from here, and in order to read this FASTQ file, you require some packages from Bioconductor or something. Okay, so what you are going to for this FASTQ file analysis, one of the packages that you require is short read, and just type Bioconductor. Okay, short read Bioconductor, the first link. And you have to install the package. You can copy the code from here and then install it. So if you copy this code and go to the R chunk over here, and then you execute this. Okay, and then it will tell you the option. Okay, now you can see the package will be installed. So it's a package that is required to read sequencing files such as FASTQ files. So it is one of the most popular packages that every NGS analyst, they use it. And in our analysis also, we are going to use this package to read the FASTQ file and do some metrics on FASTQ file input and manipulation. Okay, so you can see it will take some time, so I'm just making it a little bit smaller. Okay, now you can see the package will be installed. So also you need another package. So until this is executed, we can go and we can write okay, it's getting executed. Now you can see that when you install this package, so if you set the echo to false, these things will not be displayed. Okay, but it's a good practice that you look into the code that you are running, what are the things that are being installed, and what kind of packages that you are installing, blah blah. So yeah, it's a good option. You keep it on for now, and while rendering, what you can do is you can switch it off and then you can render it. Okay, so the next package that you are going to install is you go to another chunk and you install the package called ggplot2. So in here you can also search. ggplot2 is for the plotting purposes. Okay, oh, it's so many options. So what you can do is you go to the install, no, not this one. I think here you can write ggplot2 bioconductor. Oh, sorry, the ggplot2 has been integrated into CRAN packages. So the basic command would be any packages that you want to install from the CRAN environment is install.packages, and you click on ggplot2. Okay, and then you execute this code. You can see that it will be automatically downloaded from the CRAN mirror, and it will be installed. Okay, now the two packages that we require to read the FASTQ files have been installed. If you want to minimize the code, you can just click this one. Oh, sorry, here, this one, this option means it goes into the next code. Okay, and you can just minimize this one. So these are the first two codes that we have installed. So basically the command would be install the required packages. Okay, downloading some packages. So these are the two codes that you need to install. Now what you can do is you start by loading the FASTQ file. So the FASTQ file that we have downloaded is from this particular website. I can put the link in the description. So I named it to sample.fastq. Okay, so in order to load the FASTQ file, first what you have to do is you have to load the library. So library and then short read. Okay, library short read means it will automatically load this library. Now also you want ggplot2. So it's a good practice to load all the libraries that you have installed at the first instance, so that you don't have to load it again and again during the code execution. So it's a good option that you first load all the packages required for your bioinformatics analysis and then move on for the code execution. Okay, so you load these two packages which is required to read FASTQ files. Now read the FASTQ file. Okay, so since I'm working from the directory where my FASTQ file is here, you can see that sample.fastq file is here. So what I can do is I can go for a variable such as FASTQ file. Okay, and then your FASTQ sample.fastq. Okay, so the variable is FASTQ file, and you load the FASTQ file from there. So if you execute this, you can see that in your environment the sample.fastq will be installed, and you can see the file size. I mean, it depends whether you are working in a new environment or something, but this will be loaded in this particular environment, global environment. Okay, sample.fastq. So if the file is not displayed here, that means it's not loaded here. So the file has to be displayed in the environment, and then your file is loaded. So basically we have loaded the file in the variable called FASTQ file. Okay, now you need to read the FASTQ file. So that's why we have installed this package called shortread. So it has a function that is called read FASTQ. So the data that we are reading these FASTQ files will be stored in another variable. So let me just write FASTQ data and then read FASTQ. Read faster or read FASTQ. Okay, this is the option, or this is the function from this package which can read your FASTQ file. So if you click this and your variable, so it should be FASTQ file. Okay, so if you click this one, so you can select and then command and enter, so your file will be loaded. If you click this chunk, the whole thing will be executed. If you select any of the, if you put the cursor on, then click command and enter. I think in Windows it should be control and enter. So that particular line will be executed, this particular. If you select this line and you want to execute this particular line, you put the cursor over here or maybe over here, or select it and then run. You can see that all the codes that you have written in that chunk will not be executed, but that particular line will be executed. Now you can see that your FASTQ data has been read by this function called read FASTQ, and your data has been loaded. Okay, now what you want to do is let me just delete this. Okay, so the next chunk, what you want to do is you want to read the quality scores. So for that, what you need is because in here we are manipulating the FASTQ files, and we try to predict the quality scores of the FASTQ file. Okay, so I give a variable name called quality scores. Okay, and then load this FASTQ data as a matrix. And to do that, the basic syntax of writing any or loading any file into a matrix is as, and then you click on quality. Okay, you put braces or the brackets, and then quality is the function from the shortread function, from the shortread library. So as quality, and then you load your FASTQ data. So one of the best option in RStudio is it can tell you, it can help you to know what type of code that you are going to write, and if the files are loaded, it can display all the file names. So if you click here FASTQ and then you keep underscore, you can see that many options have been there. So you can actually explore the options, what you want to do, what type of things that you want, if you want to load a file or if you want to load the data, load a matrix, etc. So you can see that you can load the FASTQ data over here, and then you want to load it as a matrix, so we keep the option called matrix. So automatically it will show you different types of option, because we are going to load the data as a matrix. Okay, so yeah, now if you execute the code, sorry, it should be in double quotes. Okay, so in here you have this quality score variable where you load the FASTQ data and you want to predict the quality. So if you execute this code, you can see that your quality scores have been loaded in the environment. Okay, it's executed perfectly. So now we move on, and also we insert another block. Now we need to calculate the basic statistics. Okay, here you can calculate the basic statistics. So for that we need to develop something that is called an algorithm to read the quality scores and then predict the statistics for each base inside the FASTQ file. Okay, so for that I'm going to give a variable such as n base, because we don't know how many bases are there, so we just create a variable called n base, and then each row, n row, is each row of those FASTQ data or the quality scores that has been implemented just now, that we implemented here as a matrix file. We have to read the rows of those quality scores. So it should be quality and then scores. Oh, I just named it quality. Okay, doesn't matter. I mean, you can name it. So instead of quality, I gave the name quality. My mistake, my bad. Okay. So n base, n row, quality score. Then you want to store these statistics in a data frame. That's how the R works, because R is mostly dependent. If you have a large file, large matrix, or a large Excel file or CSV file, the R can read those data as a data frame. Always, the data frame is the most important aspect of this R code, basically. So what you need to do is now quality and then stats. Okay, because you need some statistics, because we are performing the statistical analysis over here. So quality stats and then data dot frame, because everything we need in the data frame. So it will be, first will be position. You can see this option has been automatically shown, because this is from the ggplot2 package. Okay, you can press F1 for any kind of help that you require or what you are doing. Okay, so this is from the ggplot2, where we define the position is equal to 1 colon n underscore basis. Okay, so I'll tell you what does this mean. So n basis, that the first line of code that we have assigned over here is the number of rows in the quality scores matrix. In the context of FASTQ data, each row corresponds to a specific base position across all reads. So n basis represents the total number of base position covered by the reads. Okay, the next option, the quality stats. What we are going to do is we are generating a data frame from the position 1 to n basis, to the n number of bases. Okay, and then you click comma, and then if you click enter, if you want to write a very big chunk of code and you don't want to write in one line, so if you click comma and then enter, it will automatically show you, it will automatically indent. I mean, this is how the R code works. Okay, so this block of code creates a data frame called quality stats with 4 columns. One is position, you go from starting position 1 to the n basis. Next is the median. Okay, next is the lower, and one is the upper percentile. Okay, let me tell you, the position 1 colon n basis, this creates a sequence from 1 to n basis and assigns it to the position. Okay, because the position is the kind of a function that we are using from ggplot2, because we need to plot that particular command as well. So it creates or it takes up this kind of functions from different packages. Okay, so each number in this sequence represents a base position in the reads. The next thing that we are going to define is median. So that's why initially I told that you need to learn some of the basic statistics in order to do some bioinformatics analysis. Okay. Next thing, we are going for the median, and there is a function called apply. Okay, and this apply, you can see this is a basic a function to manipulate any kind of data frame. Okay, so in here what you say, you define the quality, sorry, I mistake, QULA, quality, yeah, quality score, because I defined it wrong over there. So quality scores, okay, that is 1, starting from the 1, and then you have the function of x, okay, and then median of x. I'll tell you what I'm doing. Na dot rm is equal to true. So basically what this code does is, this line calculates the median quality score for each base position. The apply function is used to apply the median function to each row or the base position of the quality scores matrix. And this NARM true is used to ignore any type of NA values in the calculations or in your FASTQ file itself. Okay. So now what you can do, okay, now what you can do is you put the comma, and then you have another line of code where you create this particular lower quartile or the 25th percentile, and then you create the 75 percentile, which is the upper version, and this is the lower version. Okay, so the next command is lower is equal to, again, the apply, because it can read from quality scores, okay, starting from the 1, and then your function, that is x, okay, and this is quantile. That one, the first one is the median, this is the 25th percentile. So the command will be x, and then probes will be 0.25, because you are doing this 25th percentile, okay, to find the value below which 25% of the data fall. Okay, and in here also you have to set up na dot rm is equal to true, right? So this is for the lower, and then you have to define the upper. Again the same, but I'm just copying this one, but it is for 75th percentile. So in here you write 75, right? And you close the brackets. Okay, so now the code is complete. So basically what it does is, I'm going to explain it again. So this chunk of the code creates a data frame, quality stats, okay, with 4 columns such as position, the median, the lower, and the upper. The position 1 to n basis, this creates a sequence from 1 to n basis, the number of bases in your FASTQ file, and assigns it to the position column. Each number in this sequence represents a base position in the reads. Now you apply the median with the which calculates the median quality of the median quality score for each base position. The apply function is used to apply the median function to each row or the base position of the quality score matrix. Okay, NARM is to ignore any type of NA values. Similar to that, you set up the lower where you calculate the 25th percentile that you set up here 0.25, and the upper is for the 75% of this is the value below which the 70% of the data fall. Now you execute this particular chunk of code. Let's see. Oh, it says n basis. Oh, here, sorry, it should be n base because I defined here n base. Okay, now you execute this code and you can see that the code has been executed successfully. So now we have performed the statistics of our FASTQ file. So next thing that you want to do is you want to plot this by using the ggplot2 function. Okay, now plot the data, something like that. Then you create another chunk R, and then the basic function is ggplot. You might find different options for this ggplot. You can design absolutely beautiful, absolutely, you know, ready to, you know, publish those kind of figures by using ggplot2. So what we have to load is here the quality stats, because in here we have created this quality stats where it contains all the median, lower and the upper percentile. So we give quality stats, okay, and then AES means aesthetics for ggplot2. So we give x is equal to position. So the x values is the position, because 1 to n basis is the position of your basis, okay, and then plus. So ggplot2 has a typical type of characteristics feature where you define the code not with the comma but plus. Okay, so you define the geom line because there are many different options for defining or you know, painting your plot. So I'm using line option over here, where again I'm setting the aesthetics where my y score will be the median score, okay, median score, and then the color, let me put it as something yellow. Okay, so again plus, again I want to make some changes. Now the geom ribbon, where AES will give you y minimum is equal to lower. So this will tell you 25th percentile, the part where 25% of the data that fall, and then your upper value. So y minimum will be the lower, okay, and y max will be your 75th percentile. Then you set up alpha is equal to 0.2, and then you fill it up with color green, for instance. Okay, again I want to make some more options. So theme, I'm going to use a minimal theme. And if you have done something wrong in your code, it will automatically show this. I mean, this is the you know, best feature of RStudio, that while writing the code only, it will say you that this code has some error. So my error is, instead of you know, equal to, I gave what do you call the dash. So I put equal to, and this wrong will be vanished. It will say that okay, it's correct. Okay, so I'm using theme minimal because I don't want to make it messy. And you can define, you can name it, name your x axis and the y axis by using xlab function, xlab, and then you can define whatever you want to put in. So I write position in read as base pairs. Okay, so plus, and then y lab. On the y axis I put something called quality scores, quality score, sorry. Okay, and also the title of the plot. So the title of the plot should be gz title. This is the function that tell you. So per base sequence quality. Okay, so this is the plotting function of ggplot2 to plot the quality statistics of your FASTQ file. So if you execute this particular code, you can see that your plot has been nicely generated over here. Okay, so let me just change it to blue or something, so that, and this one to red maybe. Okay, yeah, now you can see this is per base sequence quality. That's your title. This is the y axis, that is your quality scores that you defined over here. This is the x axis title that you define xlab. Okay, and you plot the scores as a median values in blue color, and your upper percentile and the lower percentile you define by using red color, or maybe I make it to green. Yeah, something like this. Okay, so this is how you can manipulate FASTQ files, and you don't have to install any other tools such as FastQC for that, but you can analyze in R, and you have the code. So the best thing about running the codes and knowing what you are doing, what kind of bioinformatics analysis that you are doing, is to execute line by line code and then understand what you are doing. For instance, you take this chunk of code. If you do it in FastQC, it will automatically generate a HTML file, and it will show you, different kinds of very impressive plots. But you don't know what you are doing and what you have done. What is the basic algorithm yet? I mean, of course, you can read the documentation, but when you do it, you know it. That's my take from this analysis. Okay, so now since we have done this, read the FASTQ files and define quality scores or something, you write as a header. Okay, this is what you have done until now. Now the codes have been executed really well. Now you can render the option. So if you render it, if your codes have been executed perfectly, so it will show you a progress bar that it depends on your length of the code. I mean the files, or how big is your code and how big are your files. Those things will take time if your code is, if you have heavy files in that. So here the HTML file has been created, and if you click here, open in editor, or sorry, open in browser, you can see a very nice report of the analysis that you have done in here will be popped up over here. You see, install the required packages. We have written the code, and you can see these packages have been installed, and then you have installed the ggplot2. Load the FASTQ file, do some metrics, library short read. You can see the code has been loaded. Then you loaded the ggplot2. Here you read the files. Here you define the quality stats, and you can see while reading from here, it was a little bit confusing that how it should look, something like that. But when you see in the HTML file, it exactly. It looks really nice. Okay, and then you plot the data, and along with the plot it will show up over here. So this basically, we have done the first project, that we have downloaded the FASTQ file, we have read the FASTQ file. We don't have to rely on any other tools such as FastQC or MultiQC, something like that, but we have done everything in R, and we know that what we are doing for determining the quality of the FASTQ files. Okay, so this is it for now. Later on, maybe I will start doing the analysis for DE-Sec2 RNA sequencing analysis. I just want to give you an overview how the Quarto documentation work and how easy it is to write the codes over here. And you can share this HTML file to anybody, to your colleagues, to your students, to your professor, and also even you can publish it as a supplementary file where you load all the codes over here. And of course, you don't want to show these kind of options. You just want to show only the commands that you have done, except for the plots. You can show the plots as well, code and the plot, and rest of these packages, loading the packages, you don't need to show them. You can just set it up as echo false and it will not be displayed. Okay, so that's it for now. I'll see you in the next tutorial for the DE-Sec2 RNA sequencing package. Hope you like it. If you like it, please subscribe to my channel. And this is Pritham signing off.